Okay, so we resume the part 2 of this lecture. So I showed you the images of the bright field microscopy, dark field microscopy, and also phase contrast microscopes, and how the technique is done. Fluorescence microscopy is the use of fluorescent dyes, which is also called fluorophore, example fluorescein, rhodamine, to illuminate or brighten components of structures or make structures in an object or specimen which is viewed under the microscope visible. Magnification is the product of magnification, objective lens magnification, and eyepiece. Eyepiece is also called ocular lens magnification. Resolution is the ability to distinguish between details of structures. For instance, if you have a slide in front of you, you are looking for malaria parasite or an intestinal parasite, then the resolution of the microscope is very important. The effective resolution of light microscope is at 0.25 micrometers, and that's the formula there. G is equal to lambda over 2 Na. The formula is not important for you, please. So this is the bright field, dark field, and phase contrast microscope objects or specimen that I showed you. So let's look at the fluorescence. I've explained the fluorescence. So you see this one over here, this one and this one are Mycobacterium tuberculosis specimen or sputum specimen. So in doing fluorescent microscopy in a district or regional hospital lab or even a teaching hospital, we use the Oramin staining technique. Oramin staining technique. The name is over here, Oramin staining technique. So that is now the gold standard for TB, tuberculosis staining. So we have other examples here of fluorescent microscope, but that this is not TB. Then this one is the former Zernesen staining. That we call it the acid fast staining. That is the old method. This one we use this TB stains. Is it methylene blue, carbofusion, then the alcohol or the colorizer. That one is different from this. This one you just purely use what? A fluorophore or fluorescent dye to stain. You can see that it's very clear than this one. I brought this one. This is not a fluorescent stain, but all the rest are fluorescent stains. A simple microscope is a microscope with one lens. Compound microscope has 2 or more lenses. Interpupillary distance is the distance between 2 eyepiece lenses or ocular lenses to observe a perfectly fused image. When you are looking inside the microscope, there's a distance between the 2 eyes, and that distance between the 2 eyes is what we call interpupillary distance. Somebody's interpupillary distance may be longer than another person's. So most of the time, if it's long, you adjust it, make it. If your own is short, then make it short. When you adjust the objective lens, make it shorter so that you can be able to see. Light microscope uses light rays to magnify objects. So light will travel. This one is an old type, so it has a mirror. Instead of light rays, it has a mirror. So this is not electronic. So light will reflect over here, then it will bounce over here into the condenser to the eye and pass through the specimen on this stage and go through the eyepiece, then end up in the go through the objective here and end up in the eyepiece lens. Light rays from the object is passed through series of lenses to produce a final image as I've described. So what is electron microscope? It's a very big microscope and works on the same principles as an optical or light microscope, but uses electrons in place of light and uses electromagnetic plates in place of glass lenses. Why are we not using glass lenses over here, but we are using electromagnetic plates? Because the light rays over here is not light rays, so is electrons, a beam of electrons, and these ones are these electrons are supposed to travel through lenses. You know what will happen? It will break the lenses, destroy them. So that's why we chose to use what? Electromagnetic plate, which is a metal, so that the electrons can easily travel through the metal. You know, electrons even cannot travel through plastic lenses and those things, but they can travel through metals, electricity. So Ernst Ruska started development of the first electron microscope in 1931, which was the transmission electron microscope. Transmission means that the electron beam travels through the specimen and causes visualization. The scanning electron microscope was also developed in 1935. There are two types: transmission electron microscope, scanning electron microscope. And the transmission was discovered first before the scanning. In 1965, the first commercial scanning electron microscope was developed by Professor Sir Charles Oatley and marketed by the Cambridge Instrument Company as the stereoscan. So this is an electron microscope here. We have two in Noguchi Memorial Institute for Medical Research, and this is electron microscope. You see, instead of us to look at the image on an objective lens, we are rather looking at the image on computer. Because if the electrons are supposed to travel through an objective lens, you see what will happen to your eyes. Can electron travel to your electricity travel to your eyes? You will see what will happen. Your eyeballs will be gone. Your eye socket will be empty. Your brain will be destroyed. So instead of reusing objective lens, we are using what? We are using a computer monitor to see what is happening inside this big microscope. This is the electron microscope. This is another one, electron microscope, and these are the objective lens. The eyepiece lens is a computer monitor, because electrons will travel through. So if electrons travel through and they get to, and you are using your eyes to watch, you can see what will happen to your eyes. You dare not. So this is very important. I may bring a question and say label the parts of the microscope, or I'll give you the parts and say that organize them. Choose the appropriate one. So you have to be careful, learn this and baba it. So this is the base of the microscope. This is the base. This is the fine adjustment knob, the small one. It makes you see the image clearer. Coarse adjustment knob. It's also called coarse focus. This is what focuses the image, but it doesn't make you see clearly. The fine one that you now press to see clearly. You rotate clockwise or anti-clockwise to see clearly. We have the arm of the microscope. That's where we carry it. So if you are carrying the microscope, you hold the arm, then one of your palm will be under the foot, under the base of the foot of the microscope. Then one hand will hold the arm. We have the tube, which connects the objective lens and these ocular or eyepiece lenses. This is the tube. Then we have the ocular lens. It's the same as objective lens. There are two over here. Revolving nose piece. This revolving nose piece on which these four objective lenses sit on. 1, 2, 3 at the back, 1 at the back, 1 at the back, so 4. Then we have objective lens itself. There are 1, 2, 3, 4, and this is the nose, revolving nose piece. We have the mechanical stage. This black thing is the stage. That's where you put the specimen. And on this stage, there is a stage clip. Stage clip is here, which will clip your specimen, your slide onto the stage. Then we have the condenser, and this is the light source. So that when the electricity is connected, light passes through the light source. Light passes through the light source into the condenser, and the condenser is supposed to what? Condense the light onto the object. It's going to condense the light, concentrate the light on the specimen or on the object, so that the objective lens can be able to pick it up and give it to the eyepiece lens. So we have function of microscope parts. So we have the base, which is support on which microscope rests, like I said. Mechanical stage is the support on which the slide rests. Fine or coarse adjustment knob, focusing and resolution. Condenser lenses, which focuses light on the specimen. It concentrates the light onto the specimen. The arm, that's where we hold. It links the objective, the eyepiece lenses, and the base of the microscope. So that's where we hold. But if you are holding the arm or the hand, you have to support your palm at the base, so that maybe if the arm breaks down or the arm is disconnected or anybody hits you, you will not, the microscope will not fall down. So you support the base of the microscope with your hand or your palm at the base. Objective lens. The objective lens magnifies the specimen. It makes the specimen bigger for you to be able to see. Principle of light microscopy. Object is placed beyond the principal focus of first objective lens, producing inverted magnified rear image within the principal focus of the second lens. Rear image of first lens becomes object of second lens to produce virtual magnified direct image. So let's see. So this is the first lens. That is the objective lens. You can see the specimen over here, which is focused. This is the principal axis of the lens. Then the perpendicular rays, the rays passing through the center of the lens are not they are not refracted, but the rays passing through the curved periphery, they are angulated. Because the periphery is angulated or is curved, they are refracted. So when they are refracted, they meet the one going through the center of the lens. Then to form an image, inverted image, and the eye sees this image as what? Magnified. It's a virtual image. It sees as magnified, but it is inverted over here, but the brain interprets it as what? An upright image. Even though on the eye is seeing it as inverted, that the brain will now interpret it as, oh, this person, the head is not on the ground and the legs are up. No, the brain will understand and say, oh, this is the correct order. Okay, even though the eye is saying that it is upside down, the brain, I'm saying it is not upside down, it is upright. So this is how you see a malaria parasite in the microscope. So how do you properly handle a microscope? Ensure the power or switch is off. Plug in the mains on microscope. Lower the stage by turning the coarse adjustment knob anti-clockwise. Mount the slide with the cover slate facing upwards. Move condenser diaphragm and lever fully to the right. Turn the nose piece until the ×4 lens clicks in position. Operate Operate the stage control until a circle of light is seen passing through the stained part of the slide. This one you watch it in the video, so you have to learn it. It's very important. Okay, so to move the microscope around, support the base like what I said with one arm and hold its arm or limb with the other. Hold the arm of the microscope with the other. Very important. You may meet some of these things in the exams. Do not put the microscope on the surface. Lift it. After using the microscope, use the following steps to pack it. Turn the nose piece to the lowest magnification. That's the nose piece, turn it to the lowest magnification, so that the objective lens will be at the lowest magnification, because the magnification we have on the objective lenses are times 4, times 10, times 40, times 100. So you go to times 4. Turn to times 4. Then you move the stage down with the coarse adjustment knob. Wind the cable below the stage and cover with the dust and cover with the proof plastic cover. You know, when you are not using the microscope, you are supposed to cover it with a plastic rubber so that dust will not enter the microscope. So the woman demonstrated all this in her presentation. How to focus the specimen, very important. Close the left eye and use the right eye to look down the eyepiece lens. Raise the stage by manipulating the coarse and fine adjustment knobs until a sharp image is seen. Now close the right eye and keep the left eye open. The first one was the left eye open and the right eye closed. Hey, the left eye closed and the right eye open. Now close the right eye and keep the left eye open. Manipulate the focusing ring of the left eye piece until sharp image is seen. Open both eyes and using your fingertips push or pull apart the two pieces until a perfectly fused image is seen. This is the interpupillary distance. Just pull it apart so that in case your head is not as big as my own, you can be able to see into the eyepiece lenses of the microscope. Do not strain your eyes or squint when looking through the microscope. You just feel free and be comfortable. Change the magnification by turning the nose piece until the times 10 objective lens clicks into position. Okay, so when we look at histology, histology is the study of what? Tissues. The scientific study of cells is called cytology. Then when you say histopathology, is the diseases associated with tissues. Then cytopathology, diseases associated with what? Cells. A group of cells with an associated function is called a tissue. We are not going to do this, we will do it in second year. So when we are processing a tissue, this is what we will do. We have the grossing, dehydration, clearing, infiltration, embedding, sectioning. You will do this in second year. So also classification of stains. We have the basic stains, acidic stains. Examples are the H and E stains, hematoxylin, eosinophil stains. We'll do this in second year, as I said. So all the best in your exams. Let's prepare on Friday, or let's prepare on Saturday or Friday for our quiz. Then we'll now meet again at the exams. All the best. Thanks for your time.